some characteristics of sodium and potassium voltage-gated ion channels that you need to know to understand this. And we introduced this on Wednesday. So voltage-gated sodium channels, right? So if you were to kind of blow up this portion of the membrane, and I showed you kind of in a video, if you were to kind of blow up this particular portion of the membrane, remember it's a membrane like any other membrane. It's got a phospholipid bilayer, and these sodium channels and potassium channels are embedded in this membrane. Another reason I want you to watch these videos is that it allows you to help visualize these in the actual plasma membrane. So if this is a sodium channel within the plasma membrane, right? So it's situated in the plasma membrane, and it's going to open when the membrane is depolarized. So one question I always used to have is, okay, I get that like this portion of the membrane is going to be depolarized because the sodium channel is opened in this portion of the membrane, but how does it initiate? How does the very first one start? And that's from these small electrical potentials that happen in the cell body, which we'll talk about. They're postsynaptic potentials. So you have to have a certain threshold of positive charges, which we would kind of sketch like a depolarization, right? A more positive voltage. You'd have to have that happening in the cell body. So that first patch of membrane, any voltage-gated sodium channels, if that threshold is surpassed, they respond by opening, right? So they're going to be allosterically modified, but instead of by like a ligand binding to them, it's a voltage change that causes them to open, right? So you get this flood of positive charges in the cell body, and that's going to open up the sodium channels in that very first patch of membrane. They're voltage-gated. That means that they open when there's a change in voltage, and in this specific case, they open when the membrane is depolarized. So the opening results in a huge influx of sodium, because you know that the driving force for sodium it's a huge driving force, and it's into the cell. Who sets up the driving force? Right, the sodium potassium ATPase pump, right? On any cell, but these cells, it's super important, right? And the sodium will rush into the cell, causing the cell to get more depolarized. So you started with this threshold potential that was a depolarization that opened up these sodium channels. Then you had, I'm going to extend the membrane so that it looks like, so then you had a bunch of sodium leaking in. Well, the next patch of membrane has a bunch of sodium channels on it, so they're going to open, and so on and so forth, right? So you get this depolarization of the membrane in that particular patch. These protein channels, these voltage-gated sodium channels, they have an extension, a three-dimensional part of a section of amino acids that is called an inactivation gate. And so those channels will be closed, then there will be a depolarization signal. They'll open, causing a pore for sodium, but then they quickly inactivate. So there's a portion of that three-dimensional protein that plugs up the protein so that it basically, it's different than being closed. It's inactivated, meaning that it doesn't matter how much more you depolarize the membrane, that inactivation gate is plugged in there. Does that make sense? So the ion channel is essentially open, but it's inactivated. So what happens, this allows for kind of some rules that sodium channel isn't going to be able to be opened again unless that inactivation gate is taken out of there. And for that to happen, the membrane has to go back to its original resting membrane potential. And we'll talk about the importance of refractory periods and why this is evolutionarily the case in neurons. Okay, the second class of voltage-gated channels that you need to know to understand an action potential are the voltage-gated potassium channels. These also are open due to depolarization. Remember, what's going to happen if you recorded, and you guys will see graphs like this, if you recorded the voltage change on the y-axis, right, you guys know that at -70, that's where you've got your resting membrane potential. If you start opening up sodium channels, what's going to happen to the membrane potential? It's going to get more positive or more negative? More positive, right? So if I start opening up sodium channels, I'm going to get more and more and more positive, right? And the more sodium that goes in, the more positive you're going to get. Well, this is time down here. Remember how I said that this would be recording the voltage that's happening on one one tiny patch of the membrane, okay, one little section of the membrane. So over time, remember that those sodium channels inactivate. So even though they open, but very briefly after they inactivate, and so you stop depolarizing. So at that point, so the sodium channels are open, and up here, and I'll go through the various steps of this particular sketch. But what happens at the top of this is not only do the sodium channels inactivate, but that's the delay that's needed for the potassium channels to open. So again, this is still depolarization, right, from the resting membrane potential. You're still way high. In fact, this can be upwards to positive 40 millivolts. I think there's been some recordings of positive 60 millivolts. So that's a complete change in polarization of the membrane, where it used to be negative on the inside, now it's negative on the outside with respect to the inside, right? So at this point, the sodium channels inactivate, so that kind of stops that upward spike. But now it's brought back down to the resting membrane potential. Why? Because those delayed positive potassium channels have opened. So at this point, potassium channels open, but they're delayed, right? They don't open. That would be kind of counterproductive if you open the sodium and the potassium channels at the same time, right? Because you'd have positive ions coming in, and at the same time you'd have positive ions coming out. So the sodium ions flood in, the sodium channels inactivate, and then simultaneously the delayed voltage-gated potassium channels open, and potassium starts rushing out. So what happens when positive charges start rushing out? You repolarize the membrane. So that swing of the action potential is exclusively due to potassium channels that have opened, and those sodium channels are inactivated. They can't open anymore. Even though technically you're still in the depolarization state, right? You're way depolarized for -70, but they're inactivated. Those sodium channels can't reactivate until you repolarize the membrane. So there is a hyperpolarization swing of the action potential where so many potassium channels open that you actually end up hyperpolarizing below the -70 mark, and then the sodium potassium ATPase will restore it for you. So this particular part of the action potential looks like that. It's not just it actually dips below -70 because so much potassium rushes out that you dip below -70. So, but all of these steps are due to, and what we're going to have you do is memorize all the steps of an action potential, but it's all, think of it as all being based on what's happening with ion channels, okay? So this just shows in blue the sodium channels and the potassium channels. It kind of contrasts the two different structures. Obviously these are just like an artist's rendition of the structure. But what's different about the voltage-gated sodium channels that guide action potentials is this inactivation gate. So think of it like a tether ball, and the ball on the end of the tether is going to plug up that pore. That pore is totally, this is the channel that opens and closes. So this one's closed, now it's open. Now it's open, but it's inactivated. You guys see how it's open, but inactivated? So in terms of sodium flux, there's no more sodium flux, but the channel three-dimensionally is open. It's just inactivated so that you can't get any more depolarization after that. This is a really important aspect of the action potential. Potassium channels don't have an inactivation gate. So that is another reason why after you have this hyperpolarization, because they just sort of fly open and then they close in terms of timing, but they don't have an inactivation gate like the sodium channels do. So the potassium channels open and close slowly. They're delayed. They're called delayed potassium channels, delayed opening potassium channels, whereas the sodium channels open and inactivate extremely rapidly. Okay, so like I wrote on the board, this is what an action potential would look like if you recorded the voltage across the membrane when an action potential was going on. Okay, so this would be in a tiny section of the membrane that contained voltage-gated sodium and potassium channels, and also contained sodium-potassium ATPases to restore the resting membrane potential. So imagine that if I were to give you this kind of sketch on an exam and I were to ask you what are the steps of an action potential, you would want to do, just like any good scientist is going to do, is to axes, right? So my husband's a real estate agent, and he has this newsletter, and he had some graphs about like recent sales in Northfield or whatever. And on the y-axis, he had something, but on the on the x-axis he had something, but on the y-axis he didn't have anything. And I was like, you didn't label your axes anyway. So scientists label our axes, right? And so you got to label your axes if this was a particular exam question. So on the y-axis, we have our membrane potential in millivolts, right? Some of you on the exam gave me like that the membrane potential was 70 volts. That'd be awesome and everything, but that'd be kind of wicked huge magnitude of voltage, okay? So it's 70 millivolts. So millivolts is the membrane potential that's recorded across the membrane if we were able to take electrodes on one side of the membrane versus the other. And then time is on the x-axis. Remember, this would be recorded across one little patch of the membrane. And you can see that the action potential is, depending on the neuron and depending on the number of sodium and potassium channels, it can be anywhere from 1 to 4 milliseconds, right? There are 1,000 milliseconds in a second, right? And so this happens incredibly quickly, which is why if you're stepping in a fire, you can pull your foot away, right, in due time. Okay, so an action potential, the definition is listed right here for you. It's a large, rapid change in membrane potential that can only occur in excitable membranes. So that would be membranes that contain these types of channels, right? That's all you need. And when we talk about muscle physiology, what you guys are going to learn is that to contract any skeletal muscle, particularly, or cardiac fiber, you have to have these voltage-gated sodium and voltage-gated potassium channels. That's how muscle contracts. So we're specifically talking about neurons, but this same action potential and these steps are identical in your skeletal muscle cells, right? That's called a muscle action potential. So an action potential isn't just defined for neurons, it's defined for any excitable membrane that contains these particular channels. So what I've done for you is I've broken down the action potential in 7 fabulous steps, okay? And the order of events is listed for you in this very busy slide, okay? So step 1, I'm going to just toggle between these two, okay? And the other thing is that this is an image from your book, and so it's just the same thing. I don't know why, I just sometimes am kind of repetitive, as you guys know. As you're crossing your eyes at me like, okay lady, just start the action potential. So step 1 is over here, right? So you're at your resting membrane potential. What you guys might want to do on your y-axis is label it, and your x-axis is label it. Just again, active learning, write it down. And then this is minus 70, okay? So you're at your resting membrane potential where the membrane is at rest. No action potential is at that spot at that moment. So the first thing that happens is that you have a slight depolarization. That could either be from the previous action potential where the sodium is kind of flooding down the membrane, or it could be in the cell body where you have these small electrical depolarizations. Whatever it is, you get a slight depolarization of the membrane. What do you think this dashed line represents? No, this represents -70. This is the resting membrane potential down here. You get a slight depolarization, and then you get this boom. That's the threshold potential, yes. And each neuron is going to have a different threshold potential, but that threshold potential is, in this it's looking like it's about, minus 50. So it's not at super dramatic, it's not like 0. So the threshold potential has to be reached in order for this massive opening of the sodium channels in the membrane. So again, step 1 is the membrane becomes slightly depolarized, either due to a previous action potential or due to a graded potential in the cell body. And that causes these sodium channels to start opening because that is the signal for them to open. Step 2 is this huge upward spike. Remember that the voltage-gated sodium channels open really quickly, unlike the potassium channels. So they fly open, sodium rushes in, and you get this huge, almost immediate, if this is time, look, it's almost an immediate depolarization of that patch of the membrane because so much sodium is rushing in. So 2 would be threshold potential is reached. It's called threshold potential. That's the particular threshold at which, that's the voltage at which biochemically these sodium channels are going to fly open. Sodium rushes into the cell and you get that upward spike of the action potential. This is a positive feedback effect because the more sodium that rushes in, the more depolarization, the more sodium channels are going to open, the more depolarization, and so on and so forth. But how come it doesn't just keep on going forever? What happens to the sodium channels? They spontaneously inactivate. Doesn't matter how much depolarization you have. It's a timing thing. They spontaneously inactivate. They open and then immediately they spontaneously inactivate. So this upward spike of the action potential is very short-lived, and that's important. Okay. So again, the first step is the membrane becomes slightly depolarized. You start opening your voltage-gated sodium channels. Sodium begins rushing into the cell. Threshold potential is released. And then you get that all or nothing up spike of the action potential. What I mean by all or nothing is that the magnitude of voltage that's reached is the same all down the membrane. It's not like this one's going to be bigger than one in this patch of the membrane. The voltage is all or nothing. You either reach threshold potential and you fire this action potential, or it's not reached and you don't. That's all or nothing, and we're going to contrast that to graded potentials, ones that can be increased or decreased, and those happen in the cell body. But these are all or nothing. So the third step is, again, you reach the TP, which is the threshold potential, that throws the rest of the sodium channels open in that membrane patch, resulting in positive feedback. The more depolarization, the more sodium channels open, the more depolarization, the more sodium channels open, but then they suddenly inactivate. And now we're at step 4. We're at the peak of the action potential. Remember, this is time on the x-axis. At the peak of the action potential, the sodium channels rapidly begin to inactivate. That inactivation gate, that tether ball, just plugs up the pore. So it doesn't matter, even though you have depolarization, they have spontaneously inactivated. Now those slow, delayed potassium channels open, and so step 4 is achieved due to both of these things. The reason you don't continue depolarizing is due to both of these aspects. Sodium channel inactivation and delayed voltage-gated potassium channels open. So when you close sodium channels and you open potassium channels, potassium is going to fly out, and you're not going to have a combating flying in of ions. So you're going to bring that action potential back down to close to resting membrane potential. So the step 5 is closing sodium channels and opening potassium channels repolarizes the membrane, brings it back down to where it used to be. Step 6 is, since the potassium channels don't have an inactivation gate, so many of those potassium channels open that you actually end up having more potassium that leaves the cell that you end up hyperpolarizing lower than -70. So you get this hyperpolarization swing that's also known as the after hyperpolarization, meaning it's the last part of the action potential. And then step 7, how do you end up restoring that resting membrane potential? Because your sodium-potassium ATPases are just working the whole time to restore that -70 resting membrane potential. So if I were to ask you on an exam to describe the steps of the action potential, it all has to do with ion channels, right? You depolarize here, you open sodium channels. Positive feedback effect of the upswing of the action potential is step 3. Then step 4, those sodium channels inactivate spontaneously. They don't need a signal, it's a timing thing, right? They open, they inactivate. Potassium channels that are delayed open, bringing the resting membrane, or bringing the potential back to repolarizing the membrane rather. Step 6 is that you hyperpolarize because so many potassium channels open. Step 7, resting membrane potential is restored via the sodium-potassium ATPase. Incidentally, potassium channels close in this range, right? Potassium channels, they open slowly and they close slowly. So potassium channels by this time are going to be closed. The nice thing about the image from your book is that it overlays the action potential with what's happening with both sodium channels and potassium channels. So it's kind of showing you exactly what's going on with those particular voltage-gated channels in that patch of membrane. What you see overlaid below is the conductances to those ions. So do you guys see it says PNA, that's permeability to sodium. How permeable is the membrane to sodium? How permeable is the membrane to potassium? Obviously, if you open up ion channels, that suddenly increases your permeability, right? If you close those or inactivate those, you're going to lower your permeability. So you can see that the permeability of sodium at the beginning of the action potential is huge, and it's acute. That permeability immediately starts or increases, and then it immediately decreases. Whereas the permeability to potassium is much more delayed, and it ends much more slowly, right? So this is the permeability, the relative membrane permeability on the y-axis, and then again time on the x-axis, just showing you the relative membrane permeability of both of those ions. Okay, now a little bit about what we mean by all or nothing. So this would be kind of an artificial tracing. If you were able to take a neuron or other excitable membrane and you were able to put an electrode on one side of the membrane and an electrode on the other side, and you were able to artificially change the membrane potential. So you were able to depolarize it as much as you want or hyperpolarize it as much as you want. And what you would be doing is trying to monitor what happens in terms of action potentials. And what you see is that if I have like my voltmeter and I give a sub-threshold stimulus, a really small depolarization, you can see that I still can measure a depolarization, but it's sub-threshold. So those voltage-gated sodium channels are just not going to open in that positive feedback way. So if I give these sub-threshold stimuli, I'm going to get what are called graded potentials, meaning I get a little bit of depolarization. Maybe if I turn up the threshold or turn up the stimulus, I get a little bit more, a little bit more. But you have to have what's called a threshold stimulus, enough depolarization that that's going to biochemically open up those sodium channels. So you can see that the threshold potential has to be reached. And now what you can see is it doesn't matter what the stimulus is. If you've passed the threshold, you can see that even if I ramp up my stimulus, it doesn't matter. The magnitude of the action potential doesn't change. So think through this. Why? Why isn't it that I can't increase the stimulus and get more of an action potential, a higher why can't my action potential just be way higher? What am I limited by? Think about what causes an action potential, and then think about what limits how much voltage you can get. Yeah, concentration of what?
Sodium.
Yeah, concentration of sodium ion channels, right? Like you only have so many sodium ion channels in that patch of membrane, and they're going to spontaneously inactivate, and so there's no way, once they've all reached their threshold potential, they're going to fly open, and you're going to get the same concentration, and they're going to be open for the same amount of time. So this is what we mean by all or nothing, that the magnitude of an action potential is going to be exactly the same no matter what super threshold stimulus you give. If you give a sub-threshold stimulus, meaning lower to achieve threshold, you actually you can change the magnitude, right? Because that's going to guide how much depolarization you have. But over the threshold, it doesn't matter. You can give a whomping super threshold stimulus, and you're still just going to get the same magnitude of action potential. So when we draw action potentials, or when you see this sketch for a membrane, right, we always end up drawing images like this, where really each one of these would be one of the tracings that you guys just saw, steps 1 through 7, right? And we always draw them the same magnitude. It's not like the beautiful thing about this too is that because each action potential is happening in one patch of membrane and then the next one and then the next one, because biochemically they have the same ion channels, it's not like you lose voltage as you go down the axon, right? Because you're equipped with the same ion channels. So if I get a little depolarization and it leaks down there, boom, that's going to be super threshold. That causes another action potential. Sodium leaks in and starts diffusing down to this patch of membrane, boom, you get another action potential. So you don't lose your action potential magnitude, which is wonderful. Think of the sciatic nerve and the neurons that are in your sciatic nerve and the distance, cell-wise, the distance that you have to go, right? so you don't lose the magnitude of that stimulus. So the bottom line is that action potentials are all or nothing, whereas graded potentials that are sub-threshold, they evoke graded potentials. Graded, meaning that you're going to have gradations of response. But once you hit that threshold, it's going to be the same, no matter what the super-threshold stimulus is. Okay, so why would graded potentials be important? Graded potentials are used in cell bodies to initiate action potentials, and yet they wouldn't be good for axons, because the whole point of the anatomy of a neuron is to get a signal a long distance, right? And so graded potentials, those ones where the stimulus is actually going to guide the magnitude of the response, it's not going to be good for signals that are to be sent long distances. So let's just say that we could try to do a graded stimulus down the axon, which is not the case. But here's the stimulus, right? And if I give a voltage that is going to be sub-threshold, I'm going to lose that signal because I don't have the flying open of this voltage-gated sodium channels. That signal is just going to dissipate, just like diffusion. It's going to dissipate as you go down the axon. That wouldn't be good, obviously, because by the end of the axon, you're just going to lose it. It's going to be gone. There's going to be no more depolarization. Action potentials, because they're super-threshold stimuli, you have sodium channels and potassium channels and sodium-potassium ATPases all down the membrane. It doesn't decrease. That signal does not decrease with distance. So you could be 100 miles long as an axon and still maintain, as long as you've got those voltage-gated sodium and potassium channels there, the magnitude of the signal does not decrease with distance. Kind of a beautiful evolutionary trick, right? This, I think. I don't know. So, just in summary of this, let's just call out the differences between sodium and potassium channels that are voltage gated. By the way, you got a lot of different ion channels, right? You guys read a paper about the SCN1A, right? That's another sodium channel that's not necessarily related to this. So there are a lot of different types of sodium and potassium channels, right? And actually that one may be involved in the action potential. But the point is we're talking about the sodium and potassium channels that are voltage gated, that are part of an action potential. The sodium channels rapidly open, the potassium channels open in a delayed fashion. They open slowly, as you saw in that permeability slide. The sodium channels, their opening causes further depolarization of the membrane, and so they're sort of in a positive feedback effect. They've got, and potassium channels, their opening causes a hyperpolarization, right? Their opening causes a repolarization of the membrane because potassium is rushing out. So they have totally different responses, even though their stimulus is the same, depolarization. Sodium channels have that spontaneous inactivation gate. Potassium channels don't. They open and then they close in a delayed fashion. And then sodium channels, the membrane must be repolarized for their reactivation. So you got to think of the sodium and potassium channels, remember, as our three-dimensional proteins that have three-dimensional structure and they're allosterically and covalently modified, right? And so the sodium channel, its stimulus is a depolarization. So that three-dimensional pore will open up, right? And then based on its three-dimensional structure, part of that opening is that a millisecond later or so, that inactivation gate comes whomping in there, right? Well, another part of its three-dimensional structure is that inactivation gate will stay there. If we were able to take our electrodes and just keep the membrane up at like +30, +40, the inactivation gate will just stay there. And people have done these studies, where if you patch the membrane such that you keep it at +30, the inactivation gate never comes unplugged. The minute you ramp down the voltage, that protein changes shape and the inactivation gate comes out, and the protein then is closed. Does that make sense? So the voltages are what are changing the three-dimensional shape of these proteins, okay? So this is a super key point, because we're going to talk about something called the refractory period, which ends up coming back again to haunt us when we talk about cardiovascular physiology. Yeah?
There's no leakage when the inactivation gate leaves and when the protein is, or when the pore is closing?
No, I mean, of course, these three-dimensional proteins don't have like a step 1, step 2, step 3, it's all like so instantaneous, but you can think of it this way, that when I'm closed and inactivated, and activated, that means the tether ball isn't there, right, but I'm closed. The pore is closed, right? So then you get depolarization and that opens up the pore, right, and it's open for a millisecond. And then as part of its three-dimensional structure, that tether will spontaneously plug up the hole, right? As the membrane repolarizes, that's the stimulus for this three-dimensional structure to, the inactivation gate comes out and it's closed again.
Okay.
I'm not aware of any leakage that happens like once this thing comes out, and, right? It's sort of like a melding of this thing back together. Yep. What I thought you were gonna say is there are leak channels. Remember the ones I talked about that just kind of like flap open? There are leak channels that are in all membranes, and that's why the sodium-potassium ATPase constantly has to run, even if you're not having an action potential, because you would leak out charge and reach equilibrium. So, but I got your question. Okay, so the membrane must be repolarized for those sodium channels to reactivate, and therefore for you to get another action potential. So imagine if you are, lifting weights, and you obviously need to send a lot of action potentials down an axon, right? Because you need to maintain that stimulus of the muscle. Well, you wouldn't be able to send a single more action potential down there unless the membrane behind the first one repolarized. Why? Because the sodium channels would be inactivated, right? And again, this is going to come up again with cardiovascular physiology, and the reason that we need to keep the membrane in what's called refractory so that another action potential does not happen, so that your heart doesn't beat too quickly, too close together. Okay, so what is a refractory period? So first of all, the magnitude of each action potential on a membrane is the same. We covered that. It's all or nothing, right? It doesn't matter if it's a super threshold stimulus. It doesn't matter if you give it twice the amount of super threshold. It doesn't matter. The magnitude of the action potential is always the same. But then you might ask yourself, well, how do I contract a muscle like a single second or hold a muscle contraction for a minute, right? How does that happen? It's the frequency. It's not the magnitude that changes in a neuron. It's the frequency. So if I wanted just a single muscle twitch, which is one skeletal muscle fiber that responds in some way, I would put a single action potential down this neuron and cause the biological response downstream. But obviously, most of the time, we want to contract our muscles for longer than that. And so what's happening is you're sending a high frequency of action potentials down the axon. The higher the frequency, the more response you're going to get. So it's not the magnitude of the action potential, it's the frequency of the action potentials that is going to determine the strength of the signal. I'm using muscle contraction as an example because I think we can all kind of picture that. But remember, it could be neuron-to-neuron transduction as well, right? Neuron-to-neuron communication. So there are limits to how frequently action potentials can occur, and these are called refractory periods. So you can't ever summate an action potential. You're never going to get two action potentials happening in the same patch of membrane that are going to summate on top of each other. Remember that the magnitude is always going to stay the same. So how is it that we have these refractory periods? So there's two types of refractory periods. There's an absolute refractory period, and there's a relative refractory period. And it all is dependent on your ion channels and whether they're ready to go, right? So if you remember the rule that a sodium channel, in order to be opened, right, it has to be reactivated. It can't have that tether ball in there, right? It has to be ready to go. And if the membrane is depolarized, that tether ball's in there, right? So you have to remember that rule, and then you'll be fine with refractory periods. So the absolute refractory period is defined as a period of time, so this is time, right, where no amount of externally applied depolarization can initiate a second action potential in that same spot at that part of the membrane. Doesn't matter if you literally use all the power in, Niagara Falls. There's no way you're getting another action potential. So if there's an action potential at that spot, there is an absolute refractory period. Why? Because the sodium channels in that spot are inactivated, right? Ergo go, you can't get another action potential because you can't open the sodium channels. So the absolute refractory period, if we look back to our action potential slide, the action potential lasts through the falling phase of the action potential. So it starts here, right? It starts when I inactivate my sodium channels, and it lasts through kind of the falling phase. So this would be the absolute refractory period, start, and this would be kind of the end of the absolute refractory period. Notice that it's not all the way to the end of the action potential, because we'll see with relative refractory periods, there is a short period of time where if the stimulus is high enough, you actually can get another action potential. Okay? So you guys know me, I'm a mechanism person. I want you to know why. Why are you in absolute refractory period? This occurs due to, and if you want to in your notes, write mechanism, this is the mechanism. Why is the membrane unable to perform another action potential? It is because the sodium channels are inactivated at that spot, right? You know that. You're at this depolarization stage. They've already opened and sodium's flooded in, and they naturally, spontaneously inactivate. So the sodium channels are inactivated, therefore another action potential at that spot is just simply not possible. Well, absolute is, there ain't going to be another action potential, right? But relative refractory period is that small period of time towards the end of an action potential where, okay, if the superthreshold stimulus is high enough, some of my sodium channels have started to reactivate, and therefore you can get another action potential, but only if the stimulus is high enough. So again, a relative refractory period is defined as when a second stimulus can cause an action potential, but only if a higher threshold is reached. So normally we typically think of a neuron as having that established threshold potential, but during the relative refractory period, which is right here, the threshold would actually be higher. You'd have to give it a higher signal. This relative refractory period, like I said on the board, or I denoted on the board, occurs about the time of the after polarization. So if you were to sketch if I were to re-sketch this and say, okay, here I've got if that's my resting membrane potential, right? From here to here or so would be your absolute refractory period. Remember, this is time on the x-axis. And here to here would be the relative refractory period. What's the mechanism? This is due to some of the channels still being inactivated, but some of them are starting to reactivate, okay? It's going to be, remember this is a fraction of a second, and so some of them are going to start changing their conformation and getting ready to be activated again, but some of them aren't. So you would have to have a much higher stimulus in order to get the appropriate amount of depolarization in order to get that all or nothing response. Are there any questions about refractory periods? Just remember that they're times. They're referring to lengths of time, and they are guiding whether you can have another action potential at that very same moment. And this is why you can't have one action potential immediately superimposed on another. However, you can have high frequency action potentials, but there's always going to be a delay between one versus the other. Okay. How are action potentials propagated? Well, as you know now, each action potential will result in a local depolarization in that one patch of the membrane, which spreads to the next adjacent region. Why? Because the sodium that rushes in is going to just float down in the cytoplasm, right? And that's going to start opening reactivated sodium ion channels, right? And that's going to cause flooding in of sodium, and sodium's going to rush. So think about our refractory period, and then you'll be able to answer the question of why action potentials happen in one direction, even though technically they should be able to go both directions. So you guys know this is the cell body, and the signals come from the cell body, right? Determining whether you reach threshold or not. So we'll talk more about this in a second, but you're going to get, if you get a high enough depolarization through a graded signal called a postsynaptic potential, that's gonna cause this first patch of membrane, which has voltage-gated sodium channels, to start acting, right? So you get this rushing in of sodium, right? And an action potential that results. And that sodium is going to go in both directions, right? This is a cytoplasm, it's a liquid. So sodium's gonna go this way and it's gonna go this way, correct? But I don't have sodium and potassium voltage-gated ion channels around the cell body, so I can't elicit an action potential. So where's the only place I can? In the next spot, right? Well, okay, now I've got a bunch of sodium coming in from this one, and it's gonna go in both directions too. Why can't I do another action potential? Because it's a refractory, right? So the only way that the action potential can travel is one way, right? If I were to artificially take an electrode and zap it right here and cause depolarization, isn't it true that I could make it go both ways? But that's just not how we're anatomically made, right? You can do that in the lab, but you can't do that in our bodies. We've got a cell body that's attached to, this is called